In my opinion, the origin of the human milk microbiota is not only one. There may be different sources of bacteria arising in human milk. Some of them may arise from the mouth of the baby, from the skin of the mother, but other ones may arise from the digestive tract of the mother and may imply a translocation, a physiological translocation during late pregnancy and lactation. There have been a number of studies addressing the composition of the human milk microbiota depending on the place where the samples were collected, different countries and different settings, maybe urban and rural settings, these kind of things. Most probably there may be differences, but in practice, the studies published so far are difficult to compare. So I think in the last few years there has been a number of projects, for example the INSPIRE project, which is aimed to collect samples in different countries, developed countries, developing countries, and from healthy mother-infant pairs. And then with the same protocol of collection of samples, processing of samples, DNA extraction, bioinformatic analysis, all in common, just to compare what are in different countries. And it seems that there is a common core microbiota or microbiome, but some other microbes can vary depending on the geographical site. And I mean, not only the country, but also the kind of geographical location in one country. It's not the same rural settings that urban settings. There are two ways in which we can study the functions on the human milk microbiota. One is isolating strains and then characterize in the lab. We can do in vitro analysis, we can do in silico analysis, so studying their genomes. Studying their genomes, we can know which abilities a strain may have and which abilities a strain doesn't have, because has not the genetic determinants for these functions. So we can check at individual, a strain to a strain level. And in this way, we have found a bacteria that has a strong biochemical potential because they produce some metabolites that are interesting, for example, for the baby. But also we have seen that there are different mechanisms for protections against infectious diseases, or some mechanisms that may involve some bacteria in immunomodulation, but also, for example, in neuroimmodulation. For example, some bacterial strains can participate in amino acid metabolism. So, for example, the metabolism of tryptophan leads to the production of serotonin and these kind of things, and is something that has been almost no studied to the present. So this is one approach to study strains isolated from human milk and what kind of functions they may have. The second approach, and is one approach that we like very much, is to see the functions that there is, or the functions that there are in human milk, the whole setting of functions. So you can do that through metagenomic analysis, but also through metatranscriptomics, through metabolomics approaches. So you can see the global functions that there is in human milk. And I think it's an very interesting approach. I think similarly to the gut microbiota, it's very difficult to say what is a normal microbiota, because I think normality is not defined by the name of the species that there are in the microbiota. It's defined at the end of the day on the functions that the microbiota plays. So, as you know, different women may have different human milk microbiome profiles, but all of them be normal, has a healthy breast health and also healthy infants. So I think at the end of the day, which defines a normal microbiota either a gut microbiota or a human milk microbiota, are the functions that this microbiota plays in this case, for the infant. I think so. I think we can modulate the microbiota. As you know, modulation of the microbiota is a difficult issue because we know that sometimes the microbiota has dysbiotic processes, so they can lead to disease or predisposition to disease. So there are different ways in which I think we can modulate the microbiota. We can use the diet. But I think the diet probably is the most powerful one. But at the same time, I think it's one of the most unknown approach. Of course, we can use everyday longer list of probiotics, prebiotics, symbiotics, postbiotics, all these kind of things, which, of course, the properties depend on the strain or a combination of products and has to be tested one by one. And of course, there is the transfer of complex microbiotas. I mean, for example, the human milk microbiota, you can find different microbial consortia. You can use whole microbiota consortia to provide, for example, a protective effect to the baby. Of course, this is a complex approach, but I think in the future, probably will be one of the best ways to try to modulate the human milk microbiota. In fact, we have done a couple of assays. The last one will be I hope will be published in the next month, and was performed in different countries, in European countries. And I think the most difficult thing is to find a strain that is suitable for this target. So you have to characterize the strain for properties that are very specific to mastitis, and no many strains are able to cope with all these characteristics. But at the end of the day, we have, for example, in our lab, we had 3-4 candidates that are quite nice. Of course, there is a challenge, that is the production challenge, because sometimes the better or the best strain is difficult to get a high biomass or a high stability on time. So, and these are aspects that, of course, you have to check, no, before. But I think that, of course, there are strains that can modulate the human milk microbiota and that can be useful for the prevention or even the treatment of mastitis.